Good morning, my dear friends. So, in yesterday's class, we studied the cell organelles in prokaryotic cell. In that cell envelope, we started with the cell envelope, isn't it? So, cell envelope is going to have the following important features. So, these are the major cell organelles observed in the case of prokaryotic cell. Out of this, if you take the first part with respect to entire cell structure, cell envelope, it is with the glycocalyx, cell wall and plasma membrane, just recalling of points, that is all. And we studied the what is meant by glycocalyx, what is meant by cell wall and plasma membrane in yesterday's class, isn't it? And depending upon the nature of bacteria envelopes and response to gram staining. So, gram positive and gram negative bacteria were identified, and this process or this staining procedure, it was introduced by Christian Gram, the scientist who introduced this gram staining, where crystal violet is going to be utilized. So, the gram positive bacteria is Staphylococcus. If you take one of the example, it is going to have the violet color, and Escherichia coli is the gram negative bacteria. So, gram negative bacteria will going to do not retain the gram staining, whereas gram positive bacteria will going to retain the gram stain or that crystal violet. So, this was introduced by our Christian Gram. And second one is I think mesosome, isn't it? Yeah, mesosome and chromatophores. So, membrane structures which are observed in the case of this prokaryotic cells. What is meant by mesosome? It is the infoldings of the plasma membrane, and it is maybe in the vesicle form, isn't it? Yeah, vesicle form, tubule form and lamella form. Then functions of mesosome yesterday we discussed. And chromatophores, chromato refers to colored particle, the inner infoldings of the membranes in the prokaryotic, example like cyanobacteria. So, if you make up the if you observe this one here, it is very clear that the process of that infoldings with the colored particles. So, cyanobacteria, in that we can observe the chromatophores. Chromatophores ಗಳು cyanobacteria members ಅಲ್ಲಿ ಕಂಡು ಬರುತ್ತದೆ. It is a prokaryotic cell. So, I think we discussed till here, isn't it? Yeah. The next important structure is the nucleoid with respect to our cell part in the case of prokaryotic cell. So, what is meant by nucleoid? Nucleoid means what? It is formed of non-membranous circular genomic DNA. Genomic DNA which is circular and non-membranous. So, non-membranous or membranous, can write NOUS, non-membranous circular genomic DNA. What is mean by genomic DNA? What is DNA? Genetic material, isn't it? The total set of genetic material, that is a genome, which is observed in that particular cell, and it is composed of protein also. Genomic DNA is a non-membranous structure and with the proteins, So, it is formed of non-membranous circular genomic DNA. So, it is non-membranous, it is not having any covering. So, here we can observe the presence of this nucleoid, which is with the circular genomic DNA with the our protein. So, naked anta helteve, yake it is not having any covering. Alli nucleus iga nucleus anta ಏನು ಕನ್ಸಿಡರೇಷನ್ ಆಗುತ್ತೆ, ಆ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಏನಿದೆ ಅದಕ್ಕೆ ಯಾವುದೇ ಕವರಿಂಗ್ ಇಲ್ಲ, ಯಾವುದೇ ಎನ್ವಲಪ್ ಇಲ್ಲ. ಸೊ, it is circular DNA which is with the protein, it is not covered with any kind of membrane. So, it is naked, non-membranous circular DNA with the protein, and it is single chromosome or genetic material. It is a genetic material of the cell. ವಂಶವಾಹಿಗಳು ಅಂತ ನಾವು ಹೇಳ್ತೀವಿ ಅಲ್ವಾ, ಜೀನ್ಸ್ ಅಂತ ನಾವು ಹೇಳ್ತೀವಿ. So, adenu that is the circular DNA with the protein. So, it is not having membrane, so it is called nucleoid. Then many bacteria have small circular DNA outside the genomic DNA. Along with this genomic DNA, many of the bacteria are having very small additional circular DNA which is called plasmid. Plasmid, it is a extra chromosomal material, extra genetic material, circular DNA material which is present outside the main genetic material. Idenu idu main alva, main genetic material. Adrinda horagadege cytoplasm alli additional iruvanta small circular structure, that is called it as what, plasmid. So, extra chromosomal body, extra genetic material present outer side of that nucleoid structure which is called plasmid. It gives some unique phenotypic characters. It gives some of the characters. It is responsible for the expression of some of the characters. Example, resistance to antibiotics. So, this plasmid, that additional genetic material will going to show resistance power against some of the antibiotics. Antibiotics gottide nimage alva, which will work against pathogen. So, this antibiotic resistance property is going to be exhibited by this plasmid which is present outer side of the nucleoid, and it is additional, we can say it is a additional DNA or genome, it is additional DNA, that is a plasmid outside of the genomic DNA, refers to the main genetic material present in the nucleoid. That is what the plasmid and nucleoid structure. Next is the flagella. So, nucleoid gottaitalla makkale, genomic DNA, circular, non-membranous, so naked anta helteve, with the protein. Protein is important to hold that DNA molecule in that intact area. ಆ ಏರಿಯಾದಲ್ಲಿ ಒಟ್ಟು ಇರಬಾರ್ದಲ್ಲ, ಅದಕ್ಕೋಸ್ಕರ ಅದನ್ನು ಹೋಲ್ಡ್ ಮಾಡಲಿಕ್ಕೋಸ್ಕರ ಇದೆ. ಮತ್ತು ಪ್ಲಾಸ್ಮಿಡ್ additional DNA molecule. And the second one, the next concept, it is flagella. So, what is meant by flagella? Flagella is actually, it is the locomotory organ of the prokaryotic cell. These are the thin filamentous, thin filament, ಬಹಳ thin ಆಗಿರುವಂತಹ filament, long hair like structure, extensions from the cell wall of motile bacteria. It is the extension from the cell wall of motile bacteria. Motile bacteria ಅಂದ್ರೆ ಏನು? It shows movement, motility, movement. ಯಾವ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮೂಮೆಂಟ್ ಅನ್ನು ಮಾಡುತ್ತೆ ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಇರುವಂತಹ ಲೊಕೊಮೋಟರಿ ಆರ್ಗನ್, the organ which is responsible for the locomotion which is present in that bacteria. So, their number and arrangement are varied in different bacteria. ಅಂದರೆ ಏನು? ಕೆಲವೊಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಒಂದು ಫ್ಲಾಜೆಲ್ಲ ಇರ್ಬೋದು, ಕೆಲವೊಂದರಲ್ಲಿ ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಇರ್ಬೋದು. ಸೊ, ದ ಅರೇಂಜ್ಮೆಂಟ್ ಈಗ ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಇದೆ, ಹೇಗೆ ಅರೇಂಜ್ ಆಗಿದೆ, ಯಾವ ಸೈಡಲ್ಲಿ ಇದೆ. ಸೊ, arrangement of that flagella and number of flagella also going to be changed or varied from one bacteria to another bacteria. Anyhow, the flagellum or that extension, filamentous extension from the cell wall area, it is locomotory organ. And bacterial flagellum has 3 portions. So, idu cell wall inda emerge agutte, cell wall ige attachment irutte from cell wall region and having 3 parts. ಒಂದು flagellum ಅಂತ ತಗೊಂಡ್ರೆ ಅದರಲ್ಲಿ 3 ಪಾರ್ಟ್ ಬರುತ್ತೆ. One is the filament, second one is the hook and basal body. These are the 3 parts of the one single flagellum. So, flagellum has 3 parts. So, this is the one single flagellum, and here it is having the filament. So, filament is a long thread like structure which is present in that flagellum. So, filament is the longest portion anta helabodu, longest portion and extends from the cell surface to the outside, cell surface ಇಂದ ಹೊರಗಡೆಗೆ ಅದು extension ಆಗಿರುತ್ತೆ. So, ಈಗ cell wall ಇದ್ರೆ, ಈಗ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದ್ರೆ, here we can observe the structure, that is what our flagella, and hook is present here and basal body is present here. What is this basal body? Basal body is the part of that entire flagellum which is going to have the attachment with the main cell. So, ಈಗ flagellum ಇದೆಯಲ್ಲ, ಇಲ್ಲಿ ಈ flagellum ಏನಿದೆ ಅದು ಬಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾದ ಸೆಲ್ ವಾಲ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗುವಂತಹ ಪ್ಲೇಸ್. The flagellum which is going to attach to the outer wall of the cell of that bacteria through the basal body. So, basal body, it is important for that function. So, it is important for the function of attachment of the flagellum to the main cell of the main body or main cell wall of the bacteria. So, that is the basal body. So, filament is the longest extension, it is the longest extension or portion, it is from the cell surface of the bacteria to the outer side, and this flagellum, it is important for locomotion, that is the function of flagellum. And next one, Pillai and Fimbri, the next part. So, flagella is a locomotory organ with the three portions: hook, filament and basal body. Yeah. What is meant by Pillai and Fimbri? Pillai and Fimbri. What is this structure? The next part of the prokaryotic cell. So, these are surface structures. ಏನು surface structures ಅಂದರೆ which are attached to the body surface of that cell. Surface structures that have no role in motility. They are not involved in the movement. Movement ಗೆ ಸಂಬಂಧಪಟ್ಟ structure ಅಲ್ಲ. So, they are not having any kind of role in the movement. Pillai, singular pillus. Pillai are elongated tubular structures made of special protein that is called pillin. So, if you take bacterial cell wall, the flagella ಬರುತ್ತೆ, ಅದರಲ್ಲಿ ಇರಲಿ, pillais are very small hair like structures, ಹೀಗಿದೆ ಅಲ್ವಾ. Now, coming to this one pillai, pillai or pillus, pillus refers to singular, plural pillai. Pillai are elongated tubular structures made of special protein called pillin. Pillin refers to, it is the protein present in the pillai. Pillin is the protein present in the pillai, or pillai is made up of, pillai are made up of the protein called pillin. So, it is not having any role in the movement. So, these are the pillais. Illide nodi, pillais. So, they actually emerge from the plasma membrane area. Plasma membrane area ಇಂದ ಇದು plasma membrane, this is the plasma membrane. So, from there we can observe the formation of that pillai. So, these are the pillai. Okay. So, pillai which are very small elongated structure, tubular structures made up of protein called pillin. Then what is this Fimbri? Are small bristle like fibers sprouting out of the cell. So, what is this bristle like fibers? Bristle like fiber, it is small hair like structure. Bristle refers to small hair like structure which will sprout and originate, sprouting, originating from the cell. And in some bacteria they help to attach the bacteria to rock surfaces in the streams and to the host tissues. What is the function of that Fimbri? Of course, the pillai, it is responsible, sex pillai anta ಹೇಳ್ತೇವೆ, responsible with respect to reproduction point of view. ಇದರಲ್ಲಿ ಮೆನ್ಷನ್ ಮಾಡಲಿಲ್ಲ ಅಷ್ಟು ನಾನು. ಸೊ ರಿಪ್ರೊಡಕ್ಷನ್ ಪಾಯಿಂಟ್ ಆಫ್ ವ್ಯೂ ಅಲ್ಲಿ ಇಂಪಾರ್ಟೆಂಟ್ ಪಡೆಯುತ್ತೆ ಪಿಲ್ಲೈ. ಆ್ಯಂಡ್ ಹಿಯರ್ ಫಿಂಬ್ರಿಯೆ ಇಟ್ ಈಸ್ ಫಾರ್ ದ ಅಟ್ಯಾಚ್ಮೆಂಟ್ ಟು ದ ರಾಕ್ ಸರ್ಫೇಸ್. When these bacteria are present in the streams, streams or in that river bank etc., where for the attachment of this bacteria to the rock surface, ಕಲ್ಲುಗಳಿಗೆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಂಟ್ಕೊಳ್ಳಿಕ್ಕೆ ಅಟ್ಯಾಚ್ಮೆಂಟ್ ಆಗಲಿಕ್ಕೆ ಈ ಫಿಂಬ್ರಿಯಗಳು ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಟು ದ ಹೋಸ್ಟ್ ಟಿಶ್ಯೂ. ಈಗ ಹೋಸ್ಟ್ ಬಾಡಿಗೆ ಅಟ್ಯಾಕ್ ಆದರೆ ಪ್ಯಾಥೋಜನ್ ಆಗಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಇಫ್ ಇಟ್ ಈಸ್ ಪ್ಯಾಥೋಜನ್, if it attacks on the host body, then it will going to have the attachment with the host body by using this fimbriae. So these are the small bristle like, small hair like structure which are sprouting, which are originating from the cell wall, from the cell, from the wall of the cell, and that will help in the attachment. Okay. So that is about our flagella, pillai and fimbri. Next part, it is the ribosomes. Okay. So make a note of this pillin, it is the protein, okay, of the pillai. Yeah. What is this ribosome? So ribosomes means what? ಏನಿದು ribosomes ಅಂದರೆ? They are associated with the plasma membrane of prokaryote. Ribosomes are observed in both prokaryotic and eukaryotic cell. Eukaryotic cell next nodo. So what is this ribosome in terms of prokaryotic cell? They are associated with the plasma membrane of prokaryotic cell. They are about 15 nanometer by 20 nanometer in size. That is the size of this ribosome is measured about 15 nanometer to 20 nanometer in size, that is the range, and they are made up of 2 subunit. So ribosomes alli 2 subunit ide, larger subunit and smaller subunit. In the case of our prokaryotic cell, the ribosome is of type 70S. That 70S ribosome is divided into larger 50S unit and smaller 30S unit. Smaller 30S unit. So in the case of prokaryotic cell, ribosomes are of 70S type. In the eukaryotic cell, later we will study, that is 80S type. Here 70S type, and that 70S type of ribosome is actually with the 2 subunit, one larger subunit, it is called 50S subunit, and smaller subunit, it is called 30S subunit. What is this S? It is the Svedberg unit. Svedberg unit to denote the size of that subunit. And here 30S subunit, smaller subunit, larger subunit 50S subunit. When these 2 subunits are joined together, the entire ribosome is going to be formulated and formed, and it will become active. Otherwise it is inactive. ಎಂದು ಹೇಳಿದರೆ ಈಗ 50S ಮತ್ತು 30S ಸಬ್ಯೂನಿಟ್ ಇದೆಯಲ್ಲ, ಈ 2 ಸಬ್ಯೂನಿಟ್ಗಳು ಒಟ್ಟಿಗೆ ಜಾಯಿನ್ ಆದರೆ ಎಂಟೈರ್ ರೈಬೋಸೋಮ್ ಏನು ಆಕ್ಟಿವ್. ಈಗ 2 ಸಬ್ಯೂನಿಟ್ಗಳು ಡಿಟ್ಯಾಚ್ ಆದರೆ, ಸಪರೇಟ್ ಆದರೆ ರೈಬೋಸೋಮ್ಸ್ ಏನು ಇನ್ಆಕ್ಟಿವ್. ಸೊ ಆಕ್ಟಿವ್ ನೇಚರ್ ಆಫ್ ದ ರೈಬೋಸೋಮ್ ವಿಲ್ ಬಿ ದೇರ್ ವೆನ್ ದೀಸ್ 2 subunit, larger and smaller subunits are joined together. So this joining is there whenever it is needed with respect to their function. So 30S subunit, 50S subunit. ಯಾವಾಗ ಫಂಕ್ಷನ್ ಇಲ್ವಾ ಆಗ ಈ ಯೂನಿಟ್ಗಳು ಡಿಸೋಸಿಯೇಟ್ ಆಗುತ್ತೆ, ಸಪರೇಟ್ ಆಗುತ್ತೆ. ಸೊ ಸಪರೇಟ್ ಆದಾಗ ಅದು ಇನ್ಆಕ್ಟಿವ್ ಆಗುತ್ತೆ. ದಟ್ ಈಸ್ ದ ಥಿಂಗ್. ಸೊ ಎಸ್ ರೆಫರ್ಸ್ ಟು ಸ್ವೆಡ್ಬರ್ಗ್ ಯೂನಿಟ್ ಟು ಡಿನೋಟ್ ದ ಸೈಜ್ ಆಫ್ ದ್ಯಾಟ್ ರೈಬೋಸೋಮ್. ಸೊ ಅಲ್ಲಿ ಕೊಟ್ಟಿದ್ದೇನೆ ಸೆಡಿಮೆಂಟೇಷನ್ ಕೋಯಿಫಿಷಿಯೆಂಟ್ ಆರ್ ಸ್ವೆಡ್ಬರ್ಗ್ ಯೂನಿಟ್, ಎ ಮೆಜರ್ ಆಫ್ ಡೆನ್ಸಿಟಿ ಆ್ಯಂಡ್ ಸೈಜ್. ಸೊ ಯಾವ ಡೆನ್ಸಿಟಿ, ಎಷ್ಟು ಡೆನ್ಸಿಟಿಯಲ್ಲಿದೆ ಮತ್ತು ಸೈಜಲ್ಲಿ ಯಾವ ಸೈಜಲ್ಲಿದೆ ಅದನ್ನು ಡಿನೋಟ್ ಮಾಡಲಿಕ್ಕೆ, ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡುವಂಥ ಒಂದು ಯೂನಿಟ್ ಸೆಡಿಮೆಂಟೇಷನ್ ಕೋಯಿಫಿಷಿಯೆಂಟ್ ಆರ್ ಸ್ವೆಡ್ಬರ್ಗ್ ಯೂನಿಟ್. ಸೊ ಇಲ್ಲಿ ಎಸ್ ಅಂತ ಹೇಳಿದರೆ ಅದು ನೆನಪಿಟ್ಕೊಳ್ಳಿ. ಮತ್ತು ಪ್ರೋಕ್ಯಾರಿಯೋಟಿಕ್ ರೈಬೋಸೋಮ್ಸಲ್ಲಿ ಎಷ್ಟು ಯಾವುದೆಲ್ಲ ಸಬ್ ಯೂನಿಟ್ 70S ಅಂದರೆ ಎಷ್ಟು, 50S ಆ್ಯಂಡ್ 30S. Okay. Then functions of ribosomes, very important function. Ribosomes are of the site of translation, refers to ribosomes are considered as protein factories. ಪ್ರೋಟೀನ್ ಫ್ಯಾಕ್ಟರಿ. ಏನಿದು ಪ್ರೋಟೀನ್ ಫ್ಯಾಕ್ಟರಿ ಅಂದರೆ ಏನು? ಮ್ಯಾನುಫ್ಯಾಕ್ಚರ್ ಮಾಡ್ತಾರಲ್ವಾ ಫ್ಯಾಕ್ಟ್ರಿಯಲ್ಲಿ. ಸೊ ಇಲ್ಲಿ ನಮ್ಮ ಸೆಲ್ಲಲ್ಲಿ ಒಂದು ಮ್ಯಾನುಫ್ಯಾಕ್ಚರ್ ಯೂನಿಟ್ ಇದೆ, ಒಂದು ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಮಾಡುವಂಥ ಯೂನಿಟ್ ಇದೆ, ಪ್ರೋಟೀನನ್ನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವಂಥ ಯೂನಿಟ್ ಇದೆ, ದಟ್ ಈಸ್ ವಾಟ್ ರೈಬೋಸೋಮ್ಸ್. ಸೊ ರೈಬೋಸೋಮ್ಸ್ ಆರ್ ದ ಸೈಟ್ ಆಫ್ ಟ್ರಾನ್ಸ್ಲೇಷನ್. ಇಲ್ಲಿ ಒಂದು ಟೆಕ್ನಿಕಲ್ ಟರ್ಮನ್ನು ಹಾಕುವ, ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಅಂತ. ಏನಿದು ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಅಂದರೆ, ನಾವೀಗ ನಮ್ಮ ಜೀನನ್ನು ನಾವು ತಗೊಂಡ್ರೆ, DNA ತಗೊಂಡ್ರೆ, ನಾನು ಸಿಂಪಲ್ ಆಗಿ ಹೇಳಿಬಿಡ್ತೇನೆ ಯಾಕಂದರೆ ಸೆಕೆಂಡ್ ಇಯರಲ್ಲಿ ಡಿಟೇಲ್ ಆಗಿ ಬರುತ್ತೆ, ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್, ಟ್ರಾನ್ಸ್ಲೇಷನ್, ರಿಪ್ಲಿಕೇಶನ್ ಎಕ್ಸೆಟ್ರಾ ಅಂಡರ್ ಮಾಲಿಕ್ಯುಲರ್ ಬಯಾಲಜಿ. ಸೊ ವಾಟ್ ಈಸ್ ದಿಸ್ ಟ್ರಾನ್ಸ್ಲೇಷನ್? ಇಟ್ ಈಸ್ ದ ಪ್ರಾಸೆಸ್ ಆಫ್ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್. ಪ್ರೋಟೀನ್ ಹೇಗೆ ಸಿಂಥಸಿಸ್ ಆಗಬೇಕು, ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಗಬೇಕಿದ್ರೆ ಅದಕ್ಕೆ ಬೇಕಾದಂತಹ ಕೋಡಿಂಗ್ಗಳು ಯಾವುದರಲ್ಲಿರುತ್ತೆ, ಯಾವುದರಲ್ಲಿ ಬರೆದಿರ್ತದೆ ಇಂಥದೇ ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬೇಕು ಅಂತ ಅಲ್ವಾ, ಯಾವುದರಲ್ಲಿ ರೈಟಿಂಗ್ ಫಾರ್ಮೆಟಲ್ಲಿ ಇರುತ್ತೆ, ದಟ್ ಈಸ್ ಇನ್ ದ DNA. ಸೊ DNA ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಹ್ಯಾವ್ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ದಿಸ್ mRNA. ಸೊ ದಿಸ್ ಈಸ್ ದ mRNA ಚೈನ್. ಇದು ನಿಮಗೆ ಗೊತ್ತಾಗಲಿಕ್ಕೆ ಹಾಕಿದ್ದು, ಇದೇನು ಬರಿಬೇಕಂತಿಲ್ಲ, ನಿಮಗೆ ಗೊತ್ತಾಗಲಿಕ್ಕೆ ಹಾಕಿದೆ ಅಷ್ಟೇ ಆಯಿತಾ, ಏನು ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಅಂದರೆ. ಸೊ ಇಲ್ಲಿ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಆಗುವಾಗ ನಮ್ಮ ರೈಬೋಸೋಮ್ಸ್ಗಳು ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಆಗಬೇಕಿದ್ರೆ DNA ಇಂದ RNA ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬೇಕು. ಯಾವ RNA? ದಟ್ ಈಸ್ ದ mRNA, ಮೆಸೆಂಜರ್ RNA. ಸೊ DNA ಈಸ್ ದೇರ್ ಇನ್ ದ ಸೆಲ್. ನಾವು ಆಲ್ರೆಡಿ ಹೇಳಿದ್ದೇವಲ್ವಾ, DNA ಇಸ್ ದ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್, ಮೆಜಾರಿಟಿ ಆಫ್ ಲಿವಿಂಗ್ ಸೆಲ್. ಸೊ DNA ಇಂದ mRNA ಬರಬೇಕು, ಅದಕ್ಕೇನು ಹೇಳ್ತೇವೆ? ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್. ಓಕೆ, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್. DNA ಇಂದ mRNA ಬಂತೀಗ, mRNA ಬಂದ ಮೇಲೆ ಅದು ಟ್ರಾನ್ಸ್ಲೇಷನ್ಗೆ ಒಳಪಡಬೇಕು. ಇಟ್ ವಿಲ್ ಅಂಡರ್ಗೋ ಟ್ರಾನ್ಸ್ಲೇಷನ್. ವಾಟ್ ಈಸ್ ಟ್ರಾನ್ಸ್ಲೇಷನ್? ಇಟ್ ಈಸ್ ದ ಕನ್ವರ್ಷನ್ ಆಫ್ ದ್ಯಾಟ್ ಸೀಕ್ವೆನ್ಸಸ್ ವಿಚ್ ಆರ್ ರಿಟರ್ನ್ ಇನ್ ದ mRNA ಇಂಟು ಎ ಪ್ರೋಟೀನ್ ಚೈನ್. ಸೊ ಈ ರೀತಿಯಲ್ಲಿ ಬಂದಿರುವಂತಹ ಸಿಂಗಲ್ mRNA ಚೈನ್ ಏನಿದೆ, ಅದರಿಂದ ಪ್ರೋಟೀನುಗಳು ಪ್ರೊಡ್ಯೂಸ್ ಆಗುವಂತಹ ಪ್ರಾಸೆಸ್ ಏನಿದೆ ಅದನ್ನು ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಅಂತ ಹೇಳ್ತೀವಿ. From mRNA, protein is going to be synthesized. It is a biosynthetic pathway. So where we can observe the formation of a polypeptide chain. One polypeptide chain means what? It is the chain of amino acid. Chain of amino acid. Amino acids ಗೊತ್ತಿದೆಯಲ್ಲ, protein is made up of amino acid. ಒಂದು ದೊಡ್ಡ ಒಂದು ಪ್ರೋಟೀನ್ ಆಗಬೇಕಿದ್ರೆ ಅದರಲ್ಲಿ hundreds of amino acid ಬೇಕು. ಒಂದು hundreds of amino acid ಬರಬೇಕಿದ್ರೆ ಏನು ಮಾಡಬೇಕು? That is the translation. So there will be translation process result in the production of a chain of amino acid. That chain of amino acid is termed as polypeptide chain, and that polypeptide chain is the responsible material for the manufacture of one protein. That is what the translation. So protein biosynthesis or protein synthesis that is happening in the ribosome. ನಾನು ಇಲ್ಲಿ ಯಾಕೆ ಇಷ್ಟನ್ನೆಲ್ಲ ಹೇಳಿದೆ ಅಂದರೆ, ರಾಮಾಯಣ ಹೇಳಿದೆ ಯಾಕಂದರೆ ಅದು ಏನೋ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಪ್ರಾಸೆಸ್. ನೀವು ಇಲ್ಲಿ ಬರಿಬೇಕಾದಿಷ್ಟೇ, ರೈಬೋಸೋಮ್ಸ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಟ್ರಾನ್ಸ್ಲೇಷನ್, ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್. ಸೊ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಪ್ರೋಟೀನ್ ಫ್ಯಾಕ್ಟರೀಸ್. ಗಿವ್ ರೀಸನ್. ರೈಬೋಸೋಮ್ಸ್ ಆರ್ ಕನ್ಸಿಡರ್ಡ್ ಆಸ್ ಪ್ರೋಟೀನ್ ಫ್ಯಾಕ್ಟರೀಸ್. ಗಿವ್ ರೀಸನ್. ವಾಟ್ ಇಸ್ ಆನ್ಸರ್? ಬಿಕಾಸ್ ಹಿಯರ್ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಯಾಕೆ ಆಗುತ್ತೆ? ಯಾಕಂದರೆ ಟ್ರಾನ್ಸ್ಲೇಷನಿಗೆ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಗಲಿಕ್ಕೆ ಅದು ಸೆಲ್ಲಲ್ಲಿ ಅದು ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಸೊ ಸೆವೆರಲ್ ರೈಬೋಸೋಮ್ಸ್ ಮೇ ಅಟ್ಯಾಚ್ ಟು ಎ ಸಿಂಗಲ್ mRNA. ಈಗ ಒಂದು mRNA ಚೈನ್ ಇದೆ, ಒಂದು mRNA ಚೈನಿಗೆ ಹಲವಾರು ರೈಬೋಸೋಮ್ಸ್ಗಳು ಅಟ್ಯಾಚ್ ಆಗ್ಬೋದು. ಸಿಂಗಲ್ ಪಾಯಿಂಟಲ್ಲಿ ಸಿಂಗಲ್ ಸ್ಟೆಪ್ಪಲ್ಲಿ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಕಂಟಿನ್ಯೂ ಆಗ್ಬೋದು. Then that kind of structure, it is called polyribosomes or polysomes. Am I right? Yeah, polysome. Yeah, single mRNA to form a chain called polyribosome. Several ribosomes may attach. ನಾನು ಹೇಳುವುದಕ್ಕಲ್ಲಿ ಸ್ವಲ್ಪ ಸೆಂಟೆನ್ಸ್ನ ಏನು ಸ್ಟ್ರಕ್ಚರ್ಗೆ ವ್ಯತ್ಯಾಸ ಇರ್ಬೋದು, ಅಂದರೆ ಪಾಯಿಂಟ್ ಸೇಮ್ ಇರುತ್ತೆ. ದ ವೇ ಆಫ್ ಟೀಚಿಂಗಲ್ಲಿ ಆಗೇನಾಗುತ್ತೆ, ಕೆಲವೊಮ್ಮೆ ಸೆಂಟೆನ್ಸಲ್ಲಿ ನಾವು ಹೊಸ ಸೆಂಟೆನ್ಸ್ ಆನ್ ದ ಸ್ಪಾಟ್ ಮಾಡಲಿಕ್ಕೂ ಸಾಕು, ಆಯಿತಾ. ಅದೇ ಸೆಂಟೆನ್ಸನ್ನು ಹೇಳೋದಿಲ್ಲ ಕೆಲವೊಮ್ಮೆ, ಅದಲ್ಲಿರುತ್ತೆ ಸೈಡಲ್ಲಿ. ಸೊ single mRNA is going to be attached with many ribosomes at a time, that is called polysomes or polyribosomes. So, this polyribosomes may be there, then ribosomes of a polysome translate the mRNA into protein. So, into protein. So, ribosomes of a polysome, that entire complex of ribosomes with that mRNA will going to make the entire protein synthesis. So, see here, ನಾನು ಆಗ ಹೇಳಿದ್ದೇನೆ, larger subunit, smaller subunit, they will unite together to, so as a result the activation of the ribosome will be there. So, activation of ribosome and attachment with respect to that mRNA, this is the mRNA chain, ಇಲ್ಲಿ green color ಅಲ್ಲಿ ಬರುತ್ತಲ್ಲ ಅದು mRNA chain, ಇದು ribosomes, ಇಲ್ಲಿಗ protein synthesis ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ ಅಲ್ವಾ. ಸೊ ನಮಗೆ ಲೈಟ್ ಪಿಂಕ್ ಕಲರಲ್ಲಿ ಕಾಣುತ್ತೆ. ಸೊ ಅದರ ಲೆಂತ್ ಏನಿದೆ ಜಾಸ್ತಿ ಆಗ್ತಾ ಇದೆ ಅಲ್ವಾ, ಜಾಸ್ತಿಯಾಗಿ ಲೇಟರ್ ಡಿಟ್ಯಾಚ್ ಆಗುತ್ತೆ. ಯಾಕೆ, ಅಲ್ಲಿಗೆ mRNA ಮುಗಿಯಿತು ಅಂದರೆ ಏನು, ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಸ್ಟಾಪ್ ಆಯಿತು ಅಲ್ವಾ. ಸೊ ಅದಕ್ಕೆ ಪ್ರಾಸೆಸ್ಸಲ್ಲಿದೆ, ಅದು ನಾವು ಸೆಕೆಂಡ್ ಇಯರಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡುವ, ಈಗ ಇಲ್ಲಿ ಬೇಡ. ಸೊ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಸ್ಟಾಪ್ ಆಯಿತು. ಆಗ ಸ್ಟಾಪ್ ಆದಮೇಲೆ ಏನಾಗುತ್ತೆ, ರೈಬೋಸೋಮು ಜಾಯ್ನಲ್ಲಿ ಇರುತ್ತಾ, ಲಾರ್ಜರ್ ಸ್ಮಾಲ್ ಸಬ್ ಯೂನಿಟ್, ನೋ, ಇಟ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಡಿಟ್ಯಾಚ್. Detachment means what? Ribosome will become inactive. Why inactive? Because there is no need of ribosome, because the translation stops. So allige end ಆಯಿತು. ಎಂಡ್ ಆಗುವುದಂದ್ರೆ ಏನು, ರೈಬೋಸೋಮು mRNA ಬಿಟ್ಟು ಹೊರಗೆ ಹೋಗುತ್ತೆ. ಹೊರಗೆ ಹೋಗುವಾಗ ಅದು ಅಟ್ಯಾಚ್ಮೆಂಟ್ ಮಾಡಿಕೊಂಡೇ ಹೋಗುವುದಲ್ಲ, ಲಾರ್ಜರ್ ಸಬ್ ಯೂನಿಟ್ ಸ್ಮಾಲರ್ ಸಬ್ ಯೂನಿಟ್ ಅಟ್ಯಾಚ್ ಆಗಿಕೊಂಡೇ ಹೋಗುವುದಲ್ಲ, ಅದು ಸಪರೇಟ್ ಆಗುತ್ತೆ. ಸೊ the smaller subunit, larger subunit will going to separate out and result in the end of that translation, and polypeptide chain, that is the amino acid chain, means nothing but the protein is going to be synthesized. So it is the process of translation, and the function of ribosome is the translation phenomena, and this is what protein synthesis. So it is called protein factories. Okay. And next one, it is the inclusion bodies. The next concept, so ನೆನಪಿಟ್ಟುಕೊಳ್ಳಿ ಮಕ್ಕಳೇ, 70s, 50s, 30s, translation phenomena, sorry, translation phenomena, etc. Okay. Then polyribosomes or polysome, protein factories, that is the ribosome. Next concept is the inclusion body. So inclusion body ಅಂದರೆ ಏನು? So these are non-membranous stored reserve material seen freely in the cytoplasm of prokaryotic cell. ಈಗ ಒಂದು ಪೂಲ್ ಇದೆ, ಒಂದು ಸ್ವಿಮ್ಮಿಂಗ್ ಪೂಲ್ ಇದೆ. ಸ್ವಿಮ್ಮಿಂಗ್ ಪೂಲಿಗೆ ನಾವು ಒಂದು ಬಾಲನ್ನು ಹಾಕೋದು, ಅವರ ಕವರಿಂಗ್ ಬಾಲ್ ಆರ್ ಅವರ ಫುಟ್ಬಾಲ್, ವಾಲಿಬಾಲ್ ಏನಾದರೂ ಒಂದು ಹಾಕೋದು, ಅದೇನಾಗುತ್ತೆ, ತೇಲಾಡ್ಕೊಂಡು ಇರುತ್ತಲ್ವಾ. ಅದೇ ರೀತಿಯಲ್ಲಿ ಇದು ಕೂಡ, if you take cytoplasm or matrix as a pool, these inclusion bodies are going to be there in a floating fashion here and there inside that cytoplasm. That is what the reserve material. So these are non-membranous, once again non-membranous stored reserve material seen freely in the cytoplasm. ಫ್ರೀ ಆಗಿ ಆರಾಮದಲ್ಲಿ ತೇಲಾಡ್ಕೊಂಡು ಇರುತ್ತೆ, ಸ್ವಿಮ್ ಮಾಡ್ತಾ ಇರುತ್ತೆ, of prokaryotic cell. Then example, phosphate granules. So these are the stored reserve material, stored reserve material. So this is the inclusion bodies. ನೋಡಿ ಇದೆಲ್ಲ inclusion body ಆಯ್ತಾ. ಆ್ಯಂಡ್ ಎಕ್ಸಾಂಪಲ್ ನಾವು ಹೇಳೋದಿದ್ರೆ phosphate granules, then cyanophacian granules and glycogen granules, then gas vacuole, ಇದೆಲ್ಲವೂ ಕೂಡ ಎಕ್ಸಾಂಪಲ್ ಆಗುತ್ತೆ. phosphate granules, cyanophacian granules, glycogen granules, gas vacuoles. Out of these gas vacuoles, if you take, these are found in blue green algae and purple and green photosynthetic bacteria. So blue green ಇರ್ಬೋದು, purple and green photosynthetic bacteriaದಲ್ಲಿ ಇದು ಕಂಡು ಬರುತ್ತೆ, gas vacuoles. So these are the freely moving particle or stored particle in the cytoplasm. Okay. That is what we can call it as inclusion bodies. Okay, my dear friends. So, these are the major parts observed in the case of prokaryotic cell. Okay. We started from, yes, I will go back. So, inclusion body gottaitala adu freely swimming the part of the cell which is going to be helpful with respect to storage part of the cell. Yeah. Cell envelope, mesosome and chromatophores, nucleoid, flagella, pillai and fimbriae, ribosomes and inclusion bodies. These are the major parts of a prokaryotic cell, major part of the prokaryotic cell. So, up to here it is a prokaryotic cell. In the next class we will going to study the eukaryotic cell. Okay. Thank you, my dear students. We will continue in the next class. Okay. In the next class we will going to continue. Thank you.